there, and welcome to part 2 of chapter 16, the blood. When we cut ourselves, the response by our bodies is immediate and complex. And as we learned in A&P 1, the process of tissue repair involves an inflammatory response as a result of damaged blood vessels. The response at the blood vessel level will not only bring the necessary materials for healing the cut, but the vessels will also respond to minimize blood loss. So let's quickly review the steps in tissue repair, and you might recognize these figures from chapter 4 of your textbook. The first stage of the process is the inflammatory stage. Chemicals are released by damaged tissues, which signals local blood vessels to become more permeable, allowing white blood cells, fluid, and clotting proteins to seep into the injured area. The second stage involves the laying down of granulation tissue and the restoration of vascular supply. There's a cleanup process that is occurring, and granulation tissue is eventually replaced with collagen that's produced by fibroblasts. In the final stage of repair, the epithelium has been regenerated and the underlying connective tissue is fibrosed. This area will continue to undergo restructuring until all that is left as evidence of the cut is a fibrotic scar. Let's back up a bit to the point just after the cut and focus on what is happening to those blood vessels that were damaged. The purpose of these events is to prevent excessive blood loss. The process is called hemostasis, and it is broken down into 3 steps. Your book has given a very concise description of the 3 steps of hemostasis in this figure, and the last step will be elaborated on a bit further. Be sure to watch the 2 required videos on hemostasis and blood clotting. They have excellent animations to help you visualize this process better. The first step of hemostasis is vascular spasm. It is through vasoconstriction that blood loss can be reduced significantly. This response is mediated through chemicals released by cells and direct injury to the smooth muscle within the wall of the blood vessel. The second step of the process is the formation of a platelet plug. When platelets are exposed to collagen in the vessel wall, they adhere to the fibers. Now, blood vessels are lined with endothelial cells, and these cells form a lining that is like Teflon to the platelets, and they slide easily along as pressure pushes the blood through. But if there is a break in the lining and the collagen is exposed, the platelets will aggregate and begin to form a plug in the break. The platelets also release chemicals that attract other platelets, and as other platelets join, they release more chemicals, attracting more platelets. And this is a positive feedback loop. Pretty quickly, the break is filled with the plug, but it is not fully stable. And that is where the final step comes in. And this step is called coagulation or clotting. And here's where the process gets very interesting. There are a group of 12 clotting factors or procoagulants, numbered 1 through 13, that are always present in the blood. Well, all but 1. The reason that our blood is not clotting all the time is because these factors exist in an inactivated form. Under the right circumstances, such as a break in a blood vessel, the factors are converted into their active forms and begin a cascade of reactions like a domino effect. There are 2 possible pathways that lead to the eventual formation of fibrin, which is essentially the glue that holds a clot together. Of the 2 pathways, one is much shorter and faster, and it is the initiator of the mechanism, and this is the extrinsic pathway. The second pathway has more steps, and it's slower, but it is the amplifier of the mechanism. This is the intrinsic pathway. Both pathways share the final 2 steps, and we will look at those now. Factor 2, prothrombin, will be activated to become thrombin. And thrombin will activate fibrinogen, converting it to fibrin. Thrombin will also activate factor 13, which will bind fibrin strands together. Fibrin, which in its inactive form was dissolved in the blood, and once activated it becomes an insoluble polymer. Thrombin activates 3 other factors upstream on the intrinsic pathway, amplifying its own production and the production of fibrin. This greatly enhances the process and is a positive feedback system. And so this very complex mechanism has been simplified for your study. But be aware that for future reference you may need to research these pathways more thoroughly. Let's move on now to the topic of blood types and agglutination. All cell membranes have embedded within them special marker molecules, collectively called the glycocalyx. Remember from AMP1 that the glycocalyx serves to help cells identify each other. This is called cell-to-cell recognition. The glycoproteins and glycolipids that make up the glycocalyx have certain sugars that give a cell a highly specific biological marker. Red blood cells have these markers as well. And the difference between these different markers is one sugar molecule. These markers are called antigens because they have the capacity to initiate an immune response. The antigens on a person's red blood cells may be recognized as foreign if transfused into the bloodstream of another person. The immune response that may develop causes the clumping together of the red blood cells, and this is called agglutination. And the specific red blood cell antigens are called agglutinogens. And the specific antibodies that are produced in response to the agglutinogens are called agglutinins. Now I know that all these words sound alike, but if you just compare the word antigen and agglutinogen, which basically mean the same thing, you might be able to keep them straight. So as you can see, the antibody or agglutinin binds to the cell and binds the cells to each other, immobilizing them and clumping them together. This is an important function of antibodies because it sequesters the foreign cells. However, this is not a good thing to be happening in the bloodstream. And let's take a look why. In this example, we have type A donor blood being transfused into the bloodstream of a type B recipient. The recipient has anti-A antibodies circulating within their bloodstream, which will bind to the foreign red blood cells, causing them to clump. Two major problems will arise from this. The clumped red blood cells will block smaller blood vessels, cutting off the blood flow to vital tissues. Also, these red blood cells will be attacked by the immune cells, breaking them down and freeing the hemoglobin, which will be released into the bloodstream. This hemoglobin is toxic to the kidneys. Although this sounds life-threatening, and it can be, a quick response by administering fluids and diuretics to basically flush the system out can save a person's life. When we test a patient for blood types, we test the ABO blood group and the RH group. There are 4 possible blood types in the ABO group. If a person's red blood cells have the A agglutinogen or antigen, they are a type A and will have anti-B agglutinins or antibodies in their bloodstream. If a person's red blood cells have the B agglutinogens, they are a type B and they have anti-A agglutinins in their blood. If a person's red blood cells have both the A and B agglutinogen, they are type AB and have neither A nor B antibodies. And lastly, a person whose red blood cells have neither the A or B antigen is a type O. This person has both anti-A and anti-B agglutinins circulating in their bloodstream. Some of you will be testing your own blood for blood types in labs, so make sure that you understand the positive and negative findings. Antibodies of the ABO group begin to show up around the 2nd month of life, whereas an Rh negative person will not have any anti-D or Rh antibodies unless exposed to Rh positive blood. The Rh blood group contains, among others, the D agglutinogen. An Rh positive person has D antigens on their red blood cells, and an Rh negative person does not. An Rh negative person does not normally have the anti-D antibody in their bloodstream unless they're exposed to Rh positive blood. Therefore, during the initial exposure of an Rh negative person to Rh positive blood, there is no agglutination response. However, there will be an immune response of the production of antibodies, much like how our immunity builds to a particular organism upon first exposure. For an Rh negative person who now has these antibodies, the problem comes upon second exposure to Rh positive blood. And this is the possible scenario that arises when an Rh negative mother is carrying an Rh positive fetus. During the first pregnancy, there are usually no issues. However, during labor, the mother's blood is exposed to the child's blood, and the mother's system will begin to build immunity to it. During her second pregnancy, if the fetus is Rh positive, the mother's antibodies will cross the placenta and attack the baby's red blood cells. This can cause severe anemia in the baby and is called hemolytic disease of the newborn or erythroblastosis fetalis. There is a medication that can prevent this attack, but it needs to be administered prophylactically before the second pregnancy. And that concludes the information for this video. Be sure you are ready to discuss these concepts in class, and I will see you then.